Hey, everyone. It is great to see you guys today. Everybody doing well? Having a good week so far? Past the halfway point in the week. That's always good. Noon on Wednesday, right? All right. It is good to see you all. Anybody have any questions you want to start out with? All right. If not, we will jump right in. So we are going to do a little more of that Pivot Interactives today. You guys asked for a little more detail, so we will do that. We didn't get very far last week. Let me just make sure. Okay, let me know if you cannot see where it says honors biology there. Terrific. Okay, so a couple of reminders just before we get started. We do have this time zone complication in that AIM Academy is set for Eastern time, and there's no way that I can set it for multiple different time zones. So for those of you who are on the West Coast or the middle of the country, or even another country or Alaska, that means that your due date is, your due time on the due date is midnight Eastern time. So you just have to keep that in mind. There isn't really any way I can change the due date. If I did, it would register for the next day in some of the states. So I do post everything 2 to 3 weeks in advance. So I think that gives you plenty of time. So that 3 hours shouldn't make a difference, or 2 hours or 1 hour. Just make sure it'd be a good idea to keep in mind what that due date is. And remember that I don't ever take off points for any assignments that are submitted after the due date. All right, I think I have inactivated plagiarism check for now. It was giving some students really high plagiarism reports when in fact they weren't plagiarized at all. So one example is if in one of the exams, if you copied my questions into your answer, it said that you plagiarized those. So we've got to find a solution to that before we activate it again. So we're working on that. It may be back later, but for now, just ignore it if you get a report. And speaking of exams, please make sure that you number your exam answers. Don't just give me a list of a bunch of answers with no numbers. Make sure that your numbers correspond to the question number in the exam. So if question number 4, is about, something about ecology, then make sure your question number 4, that you answer question number 4 and label your answer number 4. Okay? Any questions about any of that? All right, terrific. So we started Pivot Interactives last week, and we set up a schedule. We set up your accounts. Is there anyone here now who wasn't here last week, who wasn't here to set up that Pivot Interactives account? Okay, wonderful, wonderful. Then we can just go ahead and log in. So don't create a new class key. This is just for anybody who was not here before. So make sure that you go to pivotinteractives.com, www. I'll type it into the chat if that is helpful. And put in the username and password that you created. Yes, I should spell it correctly. There you go. So put in the username and password that you made. All right. Do not set up a new account. Do not put the class key in again because you already have your account. Hopefully you remember what the account number, what your username and password are. If you don't, then you're going to have to go through the password recovery process where they'll send it to your email and all. But I'm going to go quickly through all this. So let me load mine and show you that. So since we got through part 1, I'm going to go through part 1 real quickly now and just tell you guys what the answers are that you're going to put in, because we have to, if you didn't finish part 1, you have to finish it before you can go into part 2. So let's just do this quickly. We don't need to watch all the videos again. So number 1, the first answer was cloudy white. Click submit your answer and then move on to question number 2. The answer is the starch solution turns dark blue or black when the IKI is added. Click that one and then go on to number 3. Number 3 is it remains dark blue or black. So remains dark blue or black. They might be in a different place on yours. Let me know if I'm going too quickly. I just want to make sure we get as much time to do this as we can. Number 4, make a claim. So the claim for number 4 is going to be that the amount of starch in the test tube stayed the same. Number 5, the color changes from dark to clear. Submit your answer. Number 6 is going to be the amount of starch in the test tube decreased. Number 7, you have to pick 2 answers. So your 2 answers are going to be saliva has differences from water, and something in the saliva decreases the amount of starch in the tube. Now, the next one is a field where you have to write something. Just write your name. And then lock and continue. Okay, so now you should be able to go into number 2. It should open for you now. If anyone's doesn't open, let me know. We are going to go into part 2, which is what is the function of enzymes. So that's, let's take a look at this now. Okay, so I don't think we even started this last time. If we did, we just barely got there. So we're going to go ahead and do this one. In the last section, we determined that our body must have molecules that make things happen differently. We call these molecules enzymes. Let's explore a couple of different enzymes and see if we can see a pattern that describes their function. Okay, so number 1, we come over here and it tells us what to do. If you want to have it read to you, you can click the little speaker right there. So the first enzyme we're going to test here is amylase. You can see right here, enzyme added. We can change that and we can put yes or no. We're going to leave it at no for now. So just cancel that. So it's going to say starch and enzyme no. Now there are 10 different test tubes in the video, each containing a starch solution. The solution contains 0.1 grams of starch dissolved in 5 milliliters of water and the option to add the amylase enzyme. A sample from the reaction tube, that's over here, you're going to pick it. We won't do this. We won't see this. It was dispersed into the indicator tubes at 10-second intervals. It says 20, it should say 10. The indicator turns black in the presence of carbohydrate. A black color means carbohydrates are present. Gold means there are no carbohydrates present. Number 2, a 1% starch solution is in the test tube. IKI indicator is added. Remember, IKI turns black in the presence of starch. So we don't have an enzyme in this. We're going to observe the interactive. Starch, no enzyme. Starch, no enzyme. All right, well, let's observe, and we're going to observe what happens with the early time samples and the later time samples. So let's watch and see what happens here. Okay, so clearly the early time samples are turning black. Going to watch what happens with the later time samples. There is no change. We don't have any enzyme in there. We don't expect to change when there's no enzyme. All right, so what do we observe? Think about your answer. The early time samples are black and the later time samples are black. So that's what we're going to pick. All the time samples are dark black, blue in color. Submit your answer. That is correct. Then you just scroll down and you go on to number 3. Select 3 claims about the reaction in this trial based on your observations of the samples. So 0 of them still have starch in them. They're all black. The IKI turns black in the presence of starch. So clearly they all still have starch in them. But we need 3 claims. So the starch has been digested or none of the starch has been digested. Since it's still there, we can assume none of it has been digested. This demonstrates what would be observed if enzyme action was high or low. There's no enzyme action. We don't have any enzyme in our solution, so we know that it is low. We're going to submit our answer. All right, moving on to number 4. Change in the interactive. Click change in the interactive and add enzyme by selecting yes. So click where it says change and then click yes to add the enzyme and then load. Now we're going to observe the video. Again, we're going to see what the difference is between the early time samples and the later time samples. Remember that the enzyme amylase digests starch and the IKI is black in the presence of starch. So think to yourself, what do I expect to happen over time if there's the enzyme amylase in my solution that I'm putting in? So let's watch and see what happens. Oh, immediately we see a difference. The early time samples were darker. All right, it's over now. So now we choose here. The early time samples are black, the later time samples are yellow. Yep, that's it. And we can read the rest of them to make sure. Sometimes one is more right than the other in a multiple choice, but in this case, the early time samples are black and the later ones are yellow. Submit your answer. That's right. Now we're going to go on to number 5. Recall that the black color indicates the presence of starch, while yellow indicates the absence of starch. Identify the earliest time that indicates there is no longer starch in the sample. So we're thinking this is probably right around the 40. So you can type 40 at about 40 seconds in there and submit the answer. Okay, somewhere around there. Now we make a claim, and the claim is that there was a reaction. We can see it, and the starch has been removed by the amylase. So there was a reaction, and the starch was removed from the solution, is your correct answer. Now you submit your answer, and now we're ready to go into another one. So did we do this one last week? We didn't get this far, did we, to do the little egg? Yeah, I didn't think so. I think that's about, we stopped in the middle of the last one. Okay, now we're going to look at the enzyme pepsin. So pepsin is an enzyme that's in your stomach that helps to digest protein. So we're going to see what happens when we put this protein-rich egg white cube in a solution that has pepsin in it. All right, we will see an egg white cube which contains protein suspended in a solution with the enzyme pepsin. So first we're going to have it with no enzyme at all. We've got our molecule, that's protein, and we've got no enzyme at all. We don't expect there to be, oh, I hit the wrong one. We don't expect there to be any change in that egg white because there's nothing there to change it. There's no enzyme. So what's happening? Describe what happens to the egg. Nothing. It stays the same size. The egg hasn't changed. Okay, that's right. The next thing we're going to do now, you guys see how we're doing this? I'm just reading what to do and following the directions as we go. So I'm going to add an enzyme to the environment by changing the conditions. Change and click yes, and then load. And now we've got some, you can already see it starting to break down just by having the pepsin in there. Let's watch what happens over time. Is the egg increasing in size, remaining the same, or decreasing in size? Well, we can see it shrinking as we watch. The egg is decreasing in size. Let's submit that answer. All right, now we're going to make a claim. What do we know now? Based on what we just saw, what's our claim about the enzyme pepsin? It does not make a reaction in proteins, or it makes a reaction happen in proteins. It does. It makes a reaction happen. We can see the reaction as it is being dissolved away. Okay, that's the end of the protein and pepsin. Now let's scroll down. We'll do one more. Look at the enzyme catalase. You have to scroll down both sides. So looking at catalase, hydrogen peroxide, and catalase. Here's our setup. Looks the same as the picture. You see the hydrogen peroxide in the left test tube, and we see a gas collection tube over here. Now, the first thing we're going to do here is explore hydrogen peroxide with no enzyme. So we're going to make sure it says peroxide and no, and then we're going to watch and see what happens. So the peroxide is in this tube. Is anything happening? No. How much gas is being collected in that tube? Let's fast forward this to the end and see. Nope. You can see nothing's happening. So how much gas is being collected? 0. Put a 0 in and submit your answer. Okay, that's right. Not a whole lot of gas is formed. Now we're going to add the enzyme. So we're going to change the conditions to peroxide and yes to the enzyme. Now we're going to load it. And we're going to watch the same thing happen again. This time we're going to measure the gas that's in the collection tube. So let's watch now and see what happens with the enzyme in there. You can see that gas being collected at the top there. We'll run it through to the end, and then we will measure it. So we're going to measure it using one of these little tools in the tool chest right there, the tool section. Okay, looks like we're Our reaction's slowing down now. We're not going to get much more. We want to play it through to the end anyway. I'm going to fast forward here to the end. There we go. All right, now we have to find the ruler right here. So putting the ruler on there, now we can measure how much. How much gas was collected? 10 milliliters, 20 milliliters, 30 milliliters. Not quite 35 though. It's not quite down to the 35. So maybe 33 milliliters. Let's try that and see what it says. Yep, there is a difference between the last trial we observed when no enzyme was added. Now let's describe our observations about the catalase enzyme. All right, so that's this one right here, hydrogen peroxide and catalase. When the enzyme was added, there was not a reaction? No. When the enzyme was added, there was no difference in the amount of gas produced from the trial when there wasn't catalase? That's not true either. When the catalase was added, there was a reaction that produced gas. Submit our answer. That's right. All right, so now we've observed 3 different enzymes. We observed the amylase and the starch. That was the black test tubes that turned yellow. We've observed the pepsin and the protein. That was the little egg cube. And we've observed this one, the catalase hydrogen peroxide. These are all enzymes that appear in the human body. What pattern did we see across all of the enzymes? When enzymes are added, no reaction took place. We saw reactions. We know that's not it. When enzymes are added, a reaction happened that was not seen when no enzymes were added. Well, that's true. Let's put that, but let's read the other 2. When enzymes are added, reactions happen. This is the same as the trials where no enzyme was added. That's not it. When enzymes were added, no reaction took place. We can stop there. We know that's not true. So this one, when enzymes are added, a reaction happened that was not seen when no enzymes were added. That is the correct answer. All right, now we're going to make a claim about the function of enzymes in organisms. What do you think they do? Based on these, what do you think enzymes do? They have no function in the body? The function of enzymes is to make reactions happen, or the function of enzymes is to prevent reactions from happening. All right, we saw that the enzymes made those reactions happen. So that's our answer. Okay. Anybody have any questions about how we use pivot interactives? Is that helpful to see? Now, if we were, so you would continue on. You have to finish all of part 2 before you can go to part 3. So you can do part 3. Again, just read what you're supposed to do. Read the steps in each question. Follow along. Do everything that needs to be done. And then you would get to part 4. Do enzymes cause reactions with any molecules? You can break that down too. Now, like I said, when I assign one of these pivot interactives or when we do them here in class, there won't be all these questions. I won't have you. I just wanted to walk you through all the questions that are available so that you'd know how to do it, so that you would know what the different steps are. But I won't make you do 40 questions in order to be able to do this. And Ileana, the answer is both. There will be sometimes some of these pivot interactives. This is a new program we're just using for the first time this year. So I'm trying to figure out which ones I think are useful, and I don't want to waste your time. I know you guys have a lot of things that you're are vying for your time, and I want to make sure that what we do is valuable to you. So I want to only put ones in that I think are really going to supplement what it is that you're doing. Okay. All right. So let's close this off. And now I want to talk today about, let's get my thing here, about ecology. So we're in the section now in the book, if you're keeping up with the due dates, which I highly recommend doing. We're starting to learn about ecology. What's that word even mean? What do I mean when I say ecology? Anybody? Shmuel.
Like dealing with a broader, like a bigger picture.
Bigger picture. Okay, that's part of it. Umar?
Would it be the study of ecosystems?
That's definitely part of it. Yep, you guys are putting together a puzzle here, getting all the pieces. Ileana?
It's like studying how like organisms like interact with each other, and then also like the environment that the organisms are in.
Yep. So put all those together. And that's it is the big picture that we have here of we're not narrowing down individuals. We're looking at how a group of organisms interacts with another group of organisms or organisms of the same species, as well as how they react with their physical environment. So basically, it's how the living things interact with each other and how they interact with their environment. Take living things out of it, you don't have ecology anymore, right? All right, I have a couple jokes for you guys. A little cartoon here. I just know this is going to mess with the ecosystem. Do you ever stop and think about whether there's life out there? Is there life on other planets? If they are, and they come here, I do think it would mess with our ecosystem. Probably not coming in little flying saucers, though. All right, how about this one? What kind of fish is made of 2 sodium atoms? Tuna. Tuna. All right. Who's the new heifer? Never seen herbivore. And I like this one. What's the difference between weather and climate? Not the real difference. This is a joke. What's the difference between weather and climate? You can't weather a tree, but you can climb it. All right. All right. Moving on. Let's talk about habitats. So you've been learning about ecosystems and habitats so far this week, and you'll continue learning about it a little bit more. I think one of the fascinating things about organisms that share habitats is the way they interact with one another. And one of the, one of the coolest ways is this symbiotic relationship they can have with each other. So when we say symbiosis, what we mean is a close but long-term relationship. So not just a little quick interaction and then it's over. These are long-term relationships that 2 or more different species get into. At least one of the species has to benefit in order for it to be a symbiotic relationship. Now, the word symbiosis comes from the Greek word that means living together. So that gives you an idea there. Now, we've got 3 different kinds of symbiotic relationships. Can anybody name them? Shmuel.
There's parasites.
Yep.
And there's mutualism.
Yep.
And the third one is where one wins and the other one doesn't lose.
Yeah. So the first 2 that you mentioned, those are easy to remember. We all know what a parasite is, right? Parasites do damage to their host. You all know what the word mutual means. So if something is mutually beneficial, it benefits both people or all the people who are involved. That last one's a little hard to remember, commensalism. So that's where one benefits and the other is not affected at all. It couldn't care less. Leo, do you have a question? If your hand's still up from before. All right. So there they are. You can see all 3 of them. It's a relationship where 2 or more species live closely together. Mutually beneficial, that's mutualism. That would be a plus relationship. Parasitism, where one is hurt, but the other benefits. That's a plus minus. And then commensalism is a +0 relationship. The little one is real happy because he's benefiting. He gets the shade. The big one doesn't care that the little one is there. It doesn't help him, doesn't hurt him. That is commensalism. Yeah, I like that picture too. So let's talk about commensalism first. This is where one benefits and the other is unaffected. So a cattle egret is the bird, and as cows walk along, they raise up grasshoppers and beetles that the cattle egret then eats. Once in a while, they might sit on the back of the cow and they might get then some ticks and some flies, but that doesn't really affect the cattle in any way. Ileana.
Why doesn't that affect the cattle? Like, isn't it better for the cattle when they don't have ticks or like flies on them?
Yeah, it's more, so it isn't a primary way that they're going to get rid of the ticks and the flies. Most of the time, the cattle egret is just going to walk below. It's a very good point, though. And your point, we're going to see over and over as we go through today. It's very rare that a relationship is only commensalistic. Sometimes we just don't see it all. Sometimes there are benefits that we may have to figure out later. And this is a good example of that, where there is a little bit of mutualism going on here. Primarily, though, the cattle egret is going to benefit a lot, and the cow, probably not so much. I like this little cartoon. Barry Barnacle has a lot of time to ponder the deep meaning of commensalism on his whale ride. If I'm neither helping nor hurting the whale, do I even exist? Yeah, very deep thinker there. All right, so I found a video that breaks down these three. I think it does a pretty good job of explaining these three. I've broken it into 3 sections. So let's first watch the section on commensalism. Yeah, existential crisis. We all do, don't we? I heard say the other day, you know, one of the differences between animals and people is that animals never gaze out at the stars. Think about that. Animals never wonder what's out there, but we certainly do. All right, let's watch this.
Here in nature, plants and animals interact all the time, but it can be a lot more complicated than just one thing eating another. These relationships between 2 different organisms can be examples of what we call symbiosis. Symbiosis can be defined as any kind of a close, long-term biological interaction between 2 different organisms. These interactions can be mutualistic, commensalistic, or parasitic. Today, we're going to explore the incredible world of commensalism. This big web is the home of a female spider of the genus Nephila. In her web are BB-sized spiders called dewdrop spiders, gleaming silver droplets sprinkled about. Nephila could care less. She doesn't seem to notice them. The prey Nephila wants are the larger things that fly or jump into her web. The dewdrop, on the other hand, is there to grab the smaller prey that Nephila discards. It also likes to drink the drops of dew that collect on Nephila's silken lines. The 2 species of spiders are feeding at the same table, you might say. And that describes commensalism. Commensalism is a symbiosis in which members of one species gain benefits, while the other species is neither benefited nor harmed. In Costa Rica, army ants roam freely over the floor of the jungle, ravaging everything in their paths. Other insects either flee or are carried off for dinner at a later time. This is an opportunity that can't be passed up. Numerous species of birds, such as the wood creeper, have become specialists at following army ants just to catch the insects stirred up in the chaos. Once again, commensalism and symbiosis in action.
Okay, let's talk about Nephila, the big spider. Is there any possibility that that's mutualistic? You know, it looks commensalistic. Ileana.
Maybe somehow like the smaller spider keeps the web clean or something.
Yeah. So there's definitely at least a potential benefit there. I think a lot of times these relationships that we call commensalistic could have an element of mutualism to them. But there are some that are very clearly mutualistic. As an example here, kelp and sea otters. They both benefit from this relationship. The otters eat the sea urchins, which would like to eat the kelp. And then the kelp give an anchor for the otters while they're sleeping. They both benefit. So this is definitely going to be a mutualistic relationship. Let's watch what he has to say about mutualism.
In mutualism, 2 organisms are in a relationship in which both members benefit. A classic example of this is the relationship between plants and pollinators. It's basically a win-win situation. Pollinators, like bees and butterflies, fly from flower to flower, feasting on nectar. In the meantime, the pollen that sticks to their bodies transfers from one flower to the next. The pollinated flowers are now able to develop into fruits. The insects get food, and the plants get pollinated. Like I said, a win-win situation. In South Florida, the corky stem passion vine is protected by a nasty little ant about 1/20th of an inch long called Wasmannia, the little fire ant. The ants are attracted to the plant, which not only provides nectar in its flowers, but from small nectaries on the stems of the vine as well. These little pots are filled with a nutritious liquid for the ants. The zebra longwing is one of 3 species of butterflies that lay their eggs exclusively on passion vines. The larvae, from the time they hatch, feed voraciously on the plant. Here's where Wasmannia comes in. In some cases, Wasmannia munches into the tiny butterfly eggs, killing them in the process. At other times, the ants merely check on the eggs as if they have other plans later on. Savagely, the ants stab into the tender larvae just as they're hatching and inject venom into them. The ants get food. The plant gets protection from herbivores. That's mutualism. We find mutualism as well under the sea. The polyps of corals have green algal cells called zooxanthellae living within their tissues. The algal cells provide sugars which they make through photosynthesis, something the polyps can't do. The polyps, in turn, provide a protected place to live, an animal and a plant, benefiting each other in a close relationship.
Okay, we'll talk more about this environment later, but let's move on to talk about parasitism now. Now, of course, one is harmed. We're all familiar with parasites and what they do. The mosquitoes that bite you are examples of parasites. You're their host, they're benefiting, you are not. All right, so let's, mistletoe here is an example. Mistletoe lives off of the tree. It takes nutrients from the tree. Now, slowly it will weaken the life of the tree and maybe the tree will die. But what's important to remember is that parasites tend not to kill their host. They live in or on their host and just weaken the host. All right, so it's not a predator-prey relationship. Okay, let's watch the parasite video.
The grasses of the Everglades are sometimes attacked by a parasitic plant called love vine. The name is misleading. If we look closely, we see that this love vine has little projections that penetrate the grass stems, sucking the life out of the plant. This is a great example of parasitism. In a parasitic relationship, the parasite benefits, but the host is harmed. Almost all animals are parasitized in some way, often by several different kinds of parasites at the same time. We humans have lots of them, like these mosquitoes that are sucking my blood. A parasite like this on the outside of the body of its host is called an ectoparasite. Ticks and leeches are ectoparasites as well. It's important to note that a mosquito bite is not a defensive sting, like you might get from a wasp if you accidentally disturb its nest. No, the mosquito is simply getting a meal, and only female mosquitoes feast on blood. They require a blood meal in order to produce their eggs, which hatch into larvae, which develop into pupae, which ultimately split open, and onto the surface of the water steps a new generation of mosquitoes. And this is what makes the female mosquito an ectoparasite. She gets what she needs, my blood, and her host, in this case me, gets harmed. Man, it's itchy. Let's get out of here. In parts of the tropics, a biting mosquito may inadvertently inject the microscopic plasmodia organisms that cause malaria. The plasmodia enter the red blood cells in the bloodstream of the host. They reproduce there and cause the corpuscles to explode. The result can be a feverish death. Any parasite attacking the inside of the body of its host is called an endoparasite. The human tapeworm is another endoparasite. You could have a 30-foot worm in your intestines, holding on by means of sharp hooks protruding from its head. The worm gets the food you need, helping itself, but hurting you in the process. That's parasitism. You might think parasites are just plain nasty, but they actually play a very important role. Without parasites, their host species can very well overpopulate an ecosystem, putting too much stress on the resources.
So fortunately, tapeworms are not really a big issue anymore. So our history is filled with lots of people thinking something was a good idea when actually it wasn't. You know, leeches is one example of that. They used to use leeches to take the blood out of people. Actually, they use leeches now. Anybody know how leeches might be used right now in some medical procedures? Yeah, so they do sometimes, because what happens if a person has gangrene, gangrene is where your tissues are beginning to die. and let's say you had an injury and so you have gangrene developing in your fingers. If they put leeches on your fingers, those leeches will actually draw the blood down in, and it can re-energize that tissue. So that they're experimenting with that. It's not a common procedure, but they used to do bloodletting, where they would put these leeches on people thinking they had too much blood. You know, another medical procedure that they used to do is give people tapeworms for weight loss. So the idea here is you can eat as much as you want, and the tapeworm will eat your food, and you won't gain weight. You'll actually lose weight. So yeah, they don't do that anymore. Not a good plan. Not a good plan. All right, let's move on. So there are degrees of mutualism. And what I mean by that is sometimes both organisms benefit equally, and sometimes there's way more benefit to one organism than there is to the other. Yeah, you're right, Eleni, you're right. So one of these examples that I think is really cool, where they both really rely on each other a lot, is this acacia tree and the acacia ants. So the ants go inside, they hollow out these spines, and the queen ant goes in there and lays her eggs, and the larvae then grow up in there. Now the tree also provides nectar for these ants to be able to get some food. So the ants are definitely benefiting, but the tree benefits as well, because those ants are like little guards that protect the tree. So I want you to see, I think it's such an interesting example that I want you to see a little video about it.
Whenever a huge tree falls in the Costa Rican rainforest, a gap of sunlight is formed in the canopy. New life competes for this light, like this vine racing this sapling upward. But there's one tree that never has any competition, thanks to an incredible symbiotic relationship it shares with an ant. The swollen thorn acacia and its namesake, the acacia ant, have developed harmonious roles in each other's lives. The ant's role is as protector. If any of those vines try to steal the acacia's light, the ant security guards go to work. A few good chomps on the vine stems, and it's lights out for the vine. The ants don't stop there. This relatively huge grasshopper may think it's going to take a few bites out of the non-poisonous acacia, but the ants take a few bites out of it instead. And they throw in a few stings for good measure. In exchange for all this protection, the tree takes on the provider role. It gives the insect everything it needs in terms of food and shelter. These little nodules, or nectaries, secrete a sweet nectar for the adult ants to eat. And these brownish pods at the end of some leaves are the perfect, nutrient-packed food for the ants' developing young, called larvae. The acacia ant larvae live down hollowed-out thorns at the base of the acacia leaves. Some will eventually grow into adults with wings and will fly off to replicate this evolutionary masterpiece in a new acacia.
You could almost read the mind of that grasshopper, couldn't you? It's going like, stop it. Yeah, I like that. All right, so I have a case study that I want to do to a scenario. So you guys will see sometimes when you take, like, especially achievement tests or SAT and that kind of stuff, they'll use the term science scenario. It means the same thing as a case study. I like the term case study a little bit better, but they're the same thing. So we're going to do a case study now where I'm going to tell you about some relationships on a coral reef, and you're going to then tell me which kind of symbiosis it is. So the coral reef that we're going to be talking about here is the Ningaloo Reef off the coast of Australia. This is Australia here, and here's the Ningaloo Reef right here. So this case study is going to explore all three kinds of symbiosis in a coral reef. So first of all, let's talk about what a coral reef is. A coral reef is primarily composed of this. This is calcium carbonate, and it's secreted by these tiny marine animals called polyps. They're coral polyps. They're tiny. They're about the size of the eraser on the end of a pencil. So they're really little. They're related to jellyfish and sea anemones. They've got those stinging cells like they do, and they live in colonies. Now, this calcium carbonate that they secrete protects them from the outside. It's the same substance that forms clam shells and oyster shells and even snail shells. Calcium carbonate hardens on the outside of these animals and protects them. Well, when the coral polyps themselves die, that calcium carbonate stays behind. So the animal itself is gone, but the shell of it stays there. And that provides a foundation for new polyps to grow on. Over time, that builds up the massive reef structures that we're familiar with, that we see. So really, coral are living on the skeletons of their ancestors. That's how they develop, but it's an exoskeleton, so a little bit different. All right, so coral reefs almost always form around volcanic islands. Ileana.
When we like say the word coral, are you referring to like a group of polyps, or like what are we actually referring to?
Good question, and the answer is both. The word coral means one, or it can mean a whole reef of
Oh.
thousands and thousands and thousands. It's
Okay.
the same word. Coral, plural would be corals, but when we say coral, we often just mean the reef itself. So the dead ones and the living ones. Yeah, good question. So these reefs almost always form around a volcanic island. So there we'll have some free swimming coral larvae, and then they attach. So they're free swimming when they're larvae, but then they find somewhere and they attach, often to submerged rocks or other hard surfaces around the outside of the island. Now, as they grow and expand and form these colonies, it gets bigger and bigger. And over time, the reef is what we end up with. Now, the reef itself is made up of the calcium carbonate, but it has to be cemented together. So there's an algae and there are some sand that get in between all the pieces of the old dead corals, the calcium carbonate shells, if you will, and it cements it into place. And of course, there are lots of animals that like to live around a coral reef. So they are a very, very rich ecosystem. There are many symbiotic relationships in a coral reef. So I'm going to tell you about just a couple of them, and I want you to tell me what kind they are. We'll start with this first one because he talked about that in the video. This is one of the most well-known kinds of symbiosis on a coral reef. So this involves the coral polyp, which this is, remember, it's the size of a pencil eraser, but this is it. It's blown up really big. And the zooxanthellae algae. So right here, if we took a piece of this polyp, this tentacle, probably, sorry, from the coral, and we looked at it big, there's where those zooxanthellae algae live, right inside the cells of the coral. Now, what do they do there? They do photosynthesis. This is an animal. Coral is not a plant. It looks like a plant, but it's not. It's an animal. So it can't perform photosynthesis. Animals can't, but algae can. So this algae performs photosynthesis, producing sugars for the coral. The coral then gets that sugar and it benefits. It gets some food from the algae. The algae, in exchange, get protection. They get a place to live and they get exposure to sunlight, which they need. So what kind of symbiosis is this? So I'm sorry, what I'm talking about is a mutualism. Yeah, this would be mutualistic. Mutualistic. This is not like an endoparasite because it's not a parasitic relationship, but it would be a mutualistic relationship. They're both benefiting, and they're really both benefiting equally. This is a pretty balanced mutualism that we see here. Okay, so here's the next one. This is an algae called Crustose coralline. Crustose coralline algae, well, you can think of the coral itself, those calcium carbonate shells, those are the bricks of the reef, but no brick wall is going to stand up if it doesn't have mortar. Well, that's where this Crustose coralline algae comes in. It grows as a crust over the dead parts of the coral reef, essentially cementing all those little individual particles together along with the sand that's there. Now that helps to build and strengthen the reef. And so what does the algae get in return? It gets a place to live. It's a nice place for it to live, to grow right over the reef. Again, it's going to get sunshine from the sun that shines down through onto the reef. So what is this? Yep, this is another example of mutualism. Mutualism. All right, how about this one? This is the crab on the bottom and a sea urchin on the top. So sometimes crabs will use sea urchins for protection. They either go under or they climb on top of the spiny body of the sea urchin. Now the crab gets a pretty safe place because no predator is going to come along and take a bite of this. And the sea urchin generally doesn't care. It's unaffected by the presence of the crab. We're unable to identify any benefit that the sea urchin gets. Yep, good. That is commensalism. Commensalism. Okay, so the purple, blue right here, that would be the coral. And these green ones are called copepods. So the copepods feed on the coral polyps. They either live in or on their tissues. Sometimes they transmit pathogens to the coral. So the benefit to these copepods, they get food, they get shelter, but the coral gets harmed. It is negatively impacted. The whole structure of the reef is going to be affected. You got it, parasitism, parasitism. All right, you guys have all seen Finding Nemo, so we're all familiar with this example here. This is a clownfish, and clownfish are able to live among the tentacles of the anemone, and they're protected from it. Now, these anemone are predators. They have these stinging polyps, and they attach themselves to either rocks or the ocean floor, or in this case, a coral reef. And they just sit there waiting for another fish to come by to get close enough to be entangled in those poisonous tentacles. The toxin then from the anemone is going to paralyze the prey, and then their tentacles will guide that prey right into their mouth. The anemone is also looks like a flower, but it's not, it's an animal. So it has to eat, has to consume. Clownfish, though, are the exception here. They live and hide inside the tentacles of the anemone. So they're coated with a mucus layer that actually makes them immune to the sting of the anemone. So it's not like the anemone chooses not to sting them, it's that it's stinging and they're not affected by it. So clownfish are able to live inside those tentacles while also getting protection. Now, sea anemones don't seem to benefit or suffer from the presence of clownfish. So what do you think that is? Yeah, the question mark is good, Zoe, because this is one of those examples where things are not often as we see. It's a little more complicated than that. It looks like commensalism, but when we look deeper, there are actually some unseen benefits. So remember the zooxanthellae algae that we talked about before? Well, they actually live within the cells of the anemone as well. And they need some nutrients that they don't get from the anemone. When the clownfish excretes its waste products, when it poops, it puts ammonia and sulfur and phosphorus into the water around the anemone. And that actually will be absorbed into the anemone where it will where the zooxanthellae will make use of it. And yep, Lillian, another possibility here, the clownfish may act as bait, and it may lure other fish close enough that the anemone then can get it. So maybe it's mutualistic after all. This really goes to show there's a lot of times more than one aspect to symbiosis, and many different relationships can exist. So here's another one. This is a pearlfish right here, right there. And this is a sea cucumber. Again, both animals. So parrotfish will hide inside the body of sea cucumbers for protection. And they gain shelter by living inside the sea cucumber's body. Sea cucumber doesn't seem to be harmed or helped by the presence of the pearlfish. So what do you think this is? Commensalism, commensalism. Now, again, just because we don't see it doesn't mean there isn't some hidden benefit. You know, maybe there is. We just, for right now, it looks like this is more of a commensalistic relationship. So this is a fun one. These fish are wrasses, and they're called cleaner fish. They remove parasites from bigger fish, and they benefit both of them. So they even go inside the mouth of the cleaner of the bigger fish. So this is a relationship where they both benefit. Yeah, we've got fish dentists and then fish that want to be cleaned. So mutualism at work, right? And think about this. How do you think that big fish knows not to eat the cleaner fish? I mean, basically you got food swimming right into your mouth. How much easier can it be? Why doesn't it just eat it? Yeah, Rudra, they do. They know it helps them. They want to get cleaned. You got it, Marianne. Yep. Also, that little fish is pretty small. So this fish is actually a lot bigger than it looks in this picture. You don't eat your doctor. Very true. Very true. Yeah. So there's not a lot of energy in a tiny fish. It's not going to get a lot if it eats it. And it does get a lot if it doesn't. So the fact that these cleaners don't get eaten really shows us that the benefits of getting cleaned outweigh the tiny benefit it would get from eating that little bitty fish. Yeah. Yep. All right. Here's another one. This is one of my favorites here. This is a shrimp right here, the orange and white striped one. And this is a goby. So shrimp build burrows in the sand and then they maintain those burrows. Well, the goby go into the burrows for protection. The goby sticks its head out of the burrow, acting as a lookout, telling the shrimp when there's a predator coming. So they both benefit in this case. Now, they share a burrow, but what's really fun about these two is when they leave the burrow, the shrimp, which is not able to see very well, keeps its antenna on the fish. So it swims along, touching the fish so that it can be immediately warned of any danger that there's going to be out there. Yeah, BFFs for life. I like that. Definitely, definitely mutualism. All right, how about this one? This is an isopod right here. So isopods attach themselves to fish. Sometimes they'll attach themselves on the inside of their mouths, feeding on their blood and their body fluids, and it definitely harms the fish. So what kind of symbiosis is this? Yeah, this is parasitism. We have to remember that even though we tend to think of parasites as being bad for an ecosystem, they play an important role. Like he said in that video, they will keep a population from overgrowing. And an overgrown population is going to die in some pretty bad ways. They're going to starve. They're not going to have, the predators are going to come for them. So the parasites, they're not affecting the individual fish in a good way, but they're affecting the whole fish species in a good way. Yeah, that's it. Shmuel, exactly. They're bad for the individual. They're good for the whole species in many cases. Now, you know, guys, you got parasites living in and on you right now. We have parasitic organisms in our digestive system all the time. They're on your skin, but they're kept in check by the healthy things that are there and your own immune system. So our immune systems do a really good job of keeping the parasites down. It's only when parasites overgrow or we have too many of them that it gets to be a problem. Yeah. Yeah. Excellent. I'm seeing some good examples going by there. All right. I think that's it for today. Anybody have any questions? Oh, before we go, I wanted to introduce Marianne. She's just joining our class right now. You want to wave, Marianne, so everybody can see you? I love that beautiful background you have there. So everybody say hi to Marianne. Yeah, she'll be in class with us from now on. So if any of you have any questions, don't hesitate to contact me as always. If not, I will see you next time. Thanks for coming, everybody.
Thank
Bye.
you. Bye.